Aquí. Para mí un placer poder presentar hoy a Amparo y tenerla aquí con nosotros. Bueno, os voy a introducir un poco algunos datos de Amparo, son impresionantes. A ver, yo soy súper fan de su carrera. Amparo es ingeniera agrónoma de formación y donde estudió en la Politécnica de Valencia, donde también hizo su tesis, pero ya en el campo de la ciencia y tecnología de alimentos. Después se fue a Australia y allí se hizo experta en técnicas muy avanzadas de caracterización de materiales, muy avanzadas, con neutrones, dispersión de neutrones y tal. Y ya en 2008 vuelve al Instituto de Agroquímica y Tecnología de Alimentos, que es el IATA, que está en Valencia. Y allí pues tiene múltiples funciones. Ella es profesora de investigación, jefa del grupo Envases y es la actual directora del centro, que además es un Severo Ochoa. Así que ahora nos contará un poquito. Bueno, y vosotros preguntaréis, ¿esta mujer de qué conoce a Amparo? Uy, espérate. Bueno, pues nos conocimos de casualidad porque entrábamos al mismo tiempo en el avión, en un avión que nos llevaba a Graz, que había una conferencia muy chula de polisacáridos. Y yo dije, ay, pues esta es Amparo, pues la voy a saludar. Y allí pues ya coincidimos con otras chicas, como veis aquí, de todas mujeres además, con la Romaine, que es de Barcelona, Amparo y otras chicas del IATA de Barcelona, y pasamos allí unos buenos días y estuvimos visitando también un poco la ciudad. Graz es una ciudad pequeñita, muy chula, que se puede pasear, es muy planita. Sí, y es famosa principalmente por el Murinsel, que es una isla artificial que está en medio del río Mur, así muy divina. Y yo, que siempre digo, bueno, si sobra un poquito hay que ver un poco la ciudad. Y dije, ¿qué se puede hacer en Graz? Pues en Graz hay una atracción que se llama el tobogán. Esta es la colina en la que está el tobogán y este es el tobogán por dentro. Y esa soy yo saliendo disparada del tobogán. Son 64 metros de altura y 170 de longitud, que se recorren en aproximadamente 40 segundos. Y estas somos Amparo y yo a la salida del tobogán. Queríamos morir. Además, muy traicionero porque, aunque estás dentro de una montaña, hay un momento en que hay luz. Y entonces piensas estúpidamente que vas a salir. Y no, lo que va a tirarte es casi en vertical. Súper divertido. Y bueno, ya solo dejo la palabra a Amparo, ¿vale? Y espero que podáis disfrutar tanto como lo haré yo. Gracias.
Sí. Bueno, me han pedido que lo haga en inglés, ¿verdad? ¿No? Sí. So, because is there anyone here that doesn't understand Spanish? Okay, so I will do it in English. So, well, first of all, I'm really glad to be here. As Javier told, I was a bit afraid because I was like, oh my God, I'm coming from El Cairo, 5 hours flight. I don't know if I will arrive on time, but I will try to make it, enjoyable. So, flash news? No. No. That's not my presentation. Anyway, so first of all, I would like to thank Susanna. I had a great time with her in Graz, and for inviting me, because I really love to do science dissemination. I think it's something that it needs to be done, and I always take advantage of these kind of events to make campaign for science, but specifically for food science. Why? Because I think that it has been traditionally revealed. Why? Because of familiarity. We all eat every day, and sometimes it happened to me when they ask me, what are you doing? So I'm a scientist. Oh, what kind of science? Food science. Food science? And it's like, what's food science? No, what do you have to study about food? Everything is known about food. That's not true. So food science have evolved over history. At the beginning of the 19th century, what was the aim of food science? That people didn't die, because, you know, at that time, some people ate things that were contaminated and they died and they didn't know why. So these two guys, these are two French guys. I don't know if you know them. This is Nicolas Appert. This was the one, he's considered like the father of food science. So he was a confiteur, a French confiteur. He didn't know anything about science or anything like that, but he was kind of a creative guy. So he found out that if he put food inside a glass jar and sealed it and then heat it, then this food could be eaten for a long time. He didn't know why, but it worked. In fact, a prize was given to him because at that time they needed to transport food over long distances, and they needed this food to be stable. So they gave him some money for explaining why, how did he manage to keep this food for a long time. I'm not gonna talk you a lot about that, because otherwise I will be here telling you histories. And then, of course, this is the one that explained why the method that Appert invented, which it's that's why they called it Appertization. So this is Louis Pasteur, and he's the one that really explained so why the heat treatments worked for food preservation, so the pasteurization. Then, once they knew how to keep foods stable, so then it came the Industrial Revolution, which was along the 19th century, and then in the 20th and 21st century, the technological revolution occurred, and another advantage is in terms of analytical techniques allowed us to know more about food. So how food is structured, how this structuring of food affects our health, and why my objective in here is to show you that food science is really, really important. Well, first of all, it has a great social impact, because at the end, as I told you, what we eat, so Hippocrates already told that, so let your food be your medicine. If you eat properly, you don't need to go to the doctor. In fact, it affects health, but it also affects mental health. There are anytime more science advances showing that what you eat has a direct impact not only on your physical health, but also on your mental health. Well, in here, just to show you that sometimes instead of going forward, we are going backwards. We have the problem of overweight because of the health habits, but we still have undernourishment, which instead of going down, lately it's going up, which is something to be studied. But apart from the social impact, it also has a very high economic impact. In fact, here in Spain, it's the first industrial sector, like with huge revenues and also generating lots of jobs. So that's why it's also an important sector to look at. But it also has a great environmental impact. In fact, all the food system is responsible of 1/4 of the CO2 emissions. We know we are not sustainable, but the food system, it's one of the most contaminating ones, and that's something that also needs to be tackled. And how is the food science evolving? Well, it evolves related to the global context. So you know that we have an increasing population that we have to feed, and we have to produce foods using this impressively complex food system, because it's very, very intricate and very complex. We have some geopolitical issues that are also affecting all the food system. Of course, the climate change, increasing discrepancies. I mean, there's people that has, a few people that has lots of money, and there's even more people. There's a growing population that has limited resources, and we have to provide foods that are affordable for everyone. As I mentioned, we have this double problem of certain areas where there's undernourishment, but there's also a big problem of overweight because of changing food habits. And we have huge problems of food loss. So 1/3, it's considered that around 1/3 of all the food that we produced is lost. So we also have to do something with all this lost. And then we have this plastic contamination, plastics everywhere. And in this sense, just to show you how dramatically has increased, because it looks like plastics are with us forever, but they were only produced in 1950. But now they are everywhere you look, everywhere. We need them for almost everything. And so that generates lots of residues, and 40%, more or less 40% of all the plastic produced is devoted to packaging. So packaging is a big issue in the global plastic contamination. So what's the global challenge for the food science area? So our global challenge is to find ways to cultivate, produce, and also consume foods that are healthy, that are good for our health, but also keeping our natural resources and trying to be a bit more sustainable. This, of course, this global challenge is in line with the Sustainable Development Goals of the United Nations and with funding agencies, because at the end, we are all worried about having food for our future, but producing it in a sustainable way. So in this context, as Susanna told, I'm just gonna do a little bit of institutional propaganda. So I come from the Institute of Agrochemistry and Food Technology. Of course, we have a mission, so which, as most of CSIC institutes, is to produce high quality knowledge. One peculiar thing of our institute is that, as we are in Valencia, which is also a very rich agricultural area, we've been always interacting with industries. So we do very basic research, but we also do lots of research that can be translated to companies. And that's also one of our goals, so that the things that we do in the lab can be then can report some economical benefit or new products for the industry. We have not only more than 50, almost 60 years of trajectory, as I will show you. And I'm very glad to say that we are the first institute in the area of food science receiving the Severo Ochoa Excellence recognition. We are multidisciplinary in the center. So, because at the end, another thing that I haven't said is that food science, people is like, that's a real science. It's a very complex science because at the end you need to know about it, many different disciplines to really understand all what's going on. So there's chemistry, biology, microbiology, physics, engineering. You need to know a little bit of everything, well, or collaborate with people that knows about other disciplines to really tackle all the challenges that the food science area has. So that's a bit of history of our institute. So officially it was founded by Eduardo Primo in 1966. So next year we are doing 60 years of history. Originally we were in the center of Valencia, in the institute that is now the Biomedicine Institute of Valencia, very close to the campus that it's in the center. And then in 1995 we moved to the Parc Scientific de la Universitat, which is close to Burjasot. And it's interesting also to say, well, we have this, this is a second quartile journal, but it was originally found originally created in IATA, and it's still, the editors are still from IATA. And two different institutes also sourced from researchers from IATA. So one of plant biology, the IPMCP, and another from, so other two CSIC institutes. And as I told you, as we have quite an entrepreneurship thing in the institute, there are two spin-offs that searched, well, there are more than two, but two successful ones. So the first one is Biopolis, that was acquired a few years ago by ADM, which is one of the biggest ingredient companies in the world, and then Bioinicia, which also has quite a big success. And in 2011, our facilities were extended because we were growing. We are about 300 people at IATA. And very recently, so we applied in 2022, and from 2023, we got the Severo Ochoa grant. I have to say that, because Susana told me that you also asked for a María de Maeztu grant. We tried before, we didn't get it. And we got this Severo Ochoa grant because it was the right moment, and we convinced some of the researchers in the institute that the science we were doing was really relevant, and that we had, it's just a marketing thing. I mean, you need to sell that your science is crucial for the future of humanity. So, because the people were saying, an institute project, how are we gonna ask for a project if we are so different? Because each one we do very different things. So just take advantage of that. So we took advantage of our multidisciplinarity, saying that we could cover the science from the field to the fork. So from what you produce, so there are researchers that work more in the preservation of fresh fruits and veggies, to sensorial aspects, so to the end of the chain, so taking into account also the consumers. So within this Severo Ochoa, well, as I told you, we are multidisciplinary, but all our research is more or less compiled in these three big research programs. One dealing with food and health, another one with food safety and emerging risks, because that's an important bit. And the third one, which is growing a lot in the institute, it's about sustainable food from production to consumption, so making the food chain more sustainable. And then with the Severo Ochoa, we incorporated a fourth research program, which is a transversal one about food data science, which is incorporating all the tools all the new tools about data analysis to complement the other 3 programs and boost them. So I cannot talk about all the research that is going on in the Institute, because then we would be here for many hours. But I'm just gonna give you some a few ideas of things that are done in the Institute, which I think that are interesting. In the first research program about food and health, we have very potent groups working in microbiome. That's not my field of expertise, but it's something that it's amazing for me. There are others that works with food bioactives, and also trying to understand how food affects some metabolic illnesses, which we aim. So at the end, not all the food affects everyone the same way because of our microbiome. So one of the goals is also to develop some personalized food. In the second research program, they work both on detection, but also on different strategies to guarantee food safety. So they work understanding emerging risks in the food chain, also developing quick methods for detecting contamination, and also developing new preservation methods which keep the nutritional properties of different foods. And in the third program, there are lots of things that are being done by different groups. But as I'm giving the talk, in this program, I will just talk about my book. So some things that are being researched are related to process improvement, residual, I mean, all the residues that are generated both in the field and in the industries, how to reduce and use those residues. There's also an interest in finding new protein sources. As you may know, animal proteins, which are very rich in amino acids and from a nutritional point of view, are not very sustainable to produce. So other sources were to obtain nutritional proteins. And there's a line that it's also growing in the Institute, which is cell factory. So how to produce foods and materials using microbes. And then this fourth program about food data science, which I have to tell you that we are on our way. So there are researchers that are already using tools. In our case, for instance, that we are trying to introduce these tools, we are collaborating with mathematics and people that knows about data science. And the first thing that we need to do is to find a common language, because they speak a completely different language from us. So to understand each other. And about the strategic plan that we developed with this Severo Ochoa project, it's going to be brief, but I think that it's interesting to show you. What we wanted to do is not only to reinforce our research capacities, but also to consolidate the leadership in food science. And it's very important that you find your way in the international arena, so that what you do is also relevant from an international point of view. And of course, our strength was in this translation of findings to the industry, so that was a cornerstone in our research project. Then they give you lots of money, so you can also improve your research infrastructures, contract people that helps you in the administrative issues, which also helps a lot. And of course, improve working conditions and the environment, which I think that it's very important. I mean that to make the people in your institute feel comfortable when they go to the institute. So for instance, we have yoga 2 days a week for the people of the institute. And, well, we organize the work in 4 axes, so one more focus on the science, but also how to attract talent to the institute, to gain international visibility, and the important bit of transfer the science. So as I told you, when I was preparing this talk, I was like, how I'm gonna tell you all the things. I didn't know if to tell you more technical things, but I thought that it was more interesting just to show you some bits, and then we can discuss about them. So from this first research program, I just focused on the bits they are doing in the microbiome area. So I don't know if you know, but we are 50% humans, 50% microbia. So there's more microbial cells in our body than human cells. So at the end, what we eat is what we are feeding to our bacteria. And then the bacteria are producing metabolites that affect our health, but also our mental health. So there are lots of studies going on in this sense, which are amazing for me. As I told you, that's not my expertise, but I collaborate with people that work with microbiome because for me, it's a fantastic area. And it has been for a long time known that the Mediterranean diet was a good diet, but you know that we are shifting our consumption habits, and we are all accelerated, so life is really quick. We have to work a lot, do lots of things, and then you go to the supermarket and you buy something that is already cooked. Oh, why not? Well, because it has been demonstrated that processed foods are not good. So it's better that you spend some time in cooking your own food and consuming fresh vegetables. And there are each time more evidences that why is this fresh food better than the processed food. I don't have time to tell you, but we can discuss about that later on. But there are mechanisms explaining why ultra-processed foods increase type 2 diabetes, increase obesity, increase hypertension. And in our institute, there are several groups working in this area. Just to put you an example, so the group of Dr. Sanz patented one strain, one microbial strain that was isolated from slim individuals that helps protecting. So that's a probiotic area, which also has a big market. So how changing your microbiome can also improve somehow your health. And some successful stories in this work program with companies. So another microorganism that was patented before was commercialized, which is for improving the well-being of individuals with gluten intolerance. And as I was coming here, I thought, okay, so what's related in this work program with La Mayora? So I saw this manuscript, which I was like, oh, that's very, very interesting. So there are certain researchers at Dieta that are starting also to investigate what's the microbiome of the soil, because the microbiome of the soil also affects the microbiome of the plants that you grow. So these guys made some cool experiments isolating some strains, growing those strains, and seeing how tuning the microbiome of soil can improve production, which I think that it's a very interesting area to explore. In the second research program about food safety and emerging risk, I'm just gonna show you 2 examples of things that are being done at Dieta. So there's a group, very smart group, that is working on rapid methods for detecting both chemical and biological contaminants using immunoassays. They have a huge collection of antibodies, bioconjugates for detecting mycotoxin, fungicides, insecticides in different food products. And then there's a group that are experts on food viruses. So, because normally we are used to when there's some problem in eating contaminating food, normally it's like, you don't know what's that, it's a virus. Because there are more contaminated food with viruses than with bacteria, even though it's more well known the salmonellosis or the E. coli or things like that. So she's expert in environment characterization, but also developing strategies to guarantee food safety. And some successful examples that we had, well, this group that I told you, they have recently commercialized the first and only kit available for the assessment of patulin. Patulin is a regulated mycotoxin for which there was no immunoanalytical method available, and they licensed this patent to an international company. And then our colleague from, our virus expert during the COVID crisis, so she was the one that developed the assessment of COVID in wastewater, which is a method that was then, this protocol was implemented worldwide, and it has been quite successful. And then, as I told you, the third research program about food sustainability, there are lots of groups doing very fancy things, but I'm gonna talk about the things that we are doing. So I'm from the food packaging group, but we are like two different labs. So we are the BioFun lab. Why? Because we have lots of fun doing our research. And our main goal is to try to contribute somehow to the food chain sustainability. As I told you, the food chain is one of the most contaminating ones. And what do we do what are our main research areas? So we work a lot in valorization of biomass, as I will show you. It's very important, this is a more basic piece of research, but to understand how food is structured. Why? Because it has an impact on digestibility, on processing, but also for valorization. So if you want to develop green processes to extract a certain compound, you need to know how this compound is interacting with other compounds. As we are in the packaging group, we have a big research line on polymeric materials and sustainability, as I will show you in some examples, and we love doing innovation in food ingredients. So what's our strategy for valorizing? Because this talk was about valorization. How can we use all the rubbish that we produce? So as I told you, the first thing we do is to characterize it. So what's the composition? What's in there? So we have a new residue. What's in there? What can we valorize? How these components are interacting one with each other? How can we extract the components that we like to, that we can then use? And then, what's the structure of those components? Because that will have an impact also on the final properties of the products where we incorporate these new ingredients or these new materials. So, with which kind of residues are we working with? Well, mainly we have worked with other type of residues, but we are mainly working with aquatic biomass, agroindustrial residues, and fungal biomass. I didn't know if putting you like specific examples, but I have preferred to show you a bit of different things that we are doing. So I will go quite quickly because I have lots of slides, and then you can ask me or we can discuss. So why aquatic and more specifically marine biomass for studying? Well, first of all, because we've been working for food mainly with terrestrial plants, but marine biomass is underexploited. In fact, there are lots of funding programs now focusing on the blue thing. Why? Because we have already used all our terrestrial, so let's look for something there. There's 70% of the Earth's surface is water, so what's in there? It's largely unknown because there are lots of things in the water that we don't know anything about that. And then its cultivation and collection, it also has certain advantages because it doesn't require arable land. You don't use hydro resources, which you know that are a very valuable resource nowadays, water. And then they are less reliant in climatic factors. So we know that we have climate change, and that's affecting our terrestrial crops, but not that much. They also affect, but not that much, the marine ones. They are nowadays at industrial level, they are mostly used. I'm talking now about seaweeds or algae. They are mostly used for extracting phycocolloids. What are phycocolloids? So they are hydrocolloids, so they are carbohydrates that are massively used in the food industry as texturizing, rheology modifiers. They have lots of applications. I put you an example. So agar, alginate, I don't know if you have heard about those phycocolloids. All those are extracted from algae. Depending on the species of algae, the composition is different, the phycocolloids they contain are different, and they have different properties. So it's an amazing source to study. In fact, more than 36 million tons of phycocolloids are produced yearly, so that's a lot. But you know, around 65 to 97 of the biomass, sorry, I didn't put in, sorry, is a residue. So they extract a bit of phycocolloids, and then the rest of the algae is just a residue for the companies. So how we started to work with this? Well, that was, as everything happens in life, by chance. So I was in a meeting with different food companies, and I liked algae. So I was like, oh, I'm gonna propose some project about algae. And there was a guy that had one of the biggest companies producing agar, Hispanagar, which is in Burgos. We talked, so, oh, I'm interested in that, and I'm like, oh, so I'm gonna ask for a project in this food company. Do you want to join? And he was like, not really, because at the end, they had to invest some money, and it was like, I don't know this girl, what is doing. And I told him, look, if you want, you can be in the advisory board, because I don't know anything about algae. I'm interested in working with algae, but all your knowledge can help me in my research. So you don't have to put any money. I'm just gonna tell you what I'm doing, and you tell me if it makes sense or not. So that's when I started my relationship with a food company, which is, for me, it's something needed if you want then to translate your research into the industry. So in this first project that we had with algae, what we were interested is, okay, we know that these phycocolloids have been extracted in industry for a long time, but using lots of water, like very long processes, lots of purification steps. What would happen if I skip some of those purification steps? What would I obtain? Is this ingredient healthier? It has the same properties, it has different properties. So what we were interested in is in exploring greener extraction processes for obtaining phycocolloids, to simplify the processes, and also to see if we could do something with all the residue that was generated by the seaweed industries. So what we observed was that depending on the algae we were working with, and after extracting the phycocolloids, there was a whole world to discover. So lots of things in there, lots of proteins, other fibers, so lots of things to explore that could be used as ingredients for new food applications. We thought, okay, they could be used, they have lots of cellulose in there. They could be used as new packaging materials. Even there are certain bioactives we don't know. Let's study what bioactives are in there that can be also interesting for cosmetic or. So after this first project, in which we learned a lot, we then built a very, very big project, which is a European project that it will end next year, in which we have been doing lots of things with algae. So that's the Circalgae Consortia. It's a very big consortia. And what we proposed in this project was to do this cascade approach, so using different sequential steps to extract from very valuable things, using green methods, to less valuable things, but that can be used for other applications. And at the end, we are using different macro but also micro algae to extract ingredients that can be used in food, in feed for animals, but also in cosmetics. So it's a big consortia analyzing all the compounds that we are obtaining from different varieties of macro and microalgae in this sense. And the last project that we are also running in there is more focused on proteins. So there are certain algae that have lots of proteins that haven't been studied. So algae are not used for protein extraction. Why they are not used for protein extraction? Well, first, because proteins are interacting with other compounds, are not easy to extract. But that's the goal of this new project that we have, Upcycling, which is not only for seaweed, it's also from mushroom, which I will show you later why we are also interested in mushroom. So it's using the residues that are generated at industrial scale for extracting proteins. So characterizing what's the nutritional profile of those proteins, how is the digestibility, we don't know how, if those proteins are digestible or not, what amino acid profile they have, and things like that. So that's a coordinated project with CIAL, which is another CSIC institute in Madrid. And what we are doing is extracting the proteins, characterizing all the proteins, and using also the residues for packaging, but also evaluating some of those extracts for 3D printing applications, so if they can be 3D printed. And just some bits of all the things that we've done. I'm not gonna go into technical detail, but we do from very, very basic science, so understanding how less purified extracts affect, for instance, gel formation, because as I told you, phycocolloids are great for building gels. Gels are used, well, gels like the gelatin, that's a hydrogel, but those are made from carbohydrates. So those are carbohydrates that have the ability to structure, to make structures and hold other things. You can hold water. I will show you later that you can use them for other things. So how the presence, for instance, of proteins affect gel formation? How to explain that one gel is weaker than another gel? So depending on the applications, you want the gels with different physical properties. So we can understand how the carbohydrate chains get ordered and how the presence of other compounds affect the ordering of those, so at the nano or molecular scale, how they affect the macro physical properties of the gels. And then we have used them for lots of things. The phycocolloids are normally sulfated polysaccharides, so they are negatively charged, so they can interact with positively charged molecules like proteins, so they can form this kind of encapsulation structures. They can be used for delivery, so control delivery. So you can tune the composition of these capsules so that they can release their contents at different pH, so making resistance for the gastric pH and then releasing their contents in basic pH of the intestine, so they could be used for intestinal release. And as I told you, we have this problem of obesity. One of the food additives that are very much related with problems of obesity and other metabolic problems are saturated fats. So industry is always looking for alternatives to saturated fats. And we thought, okay, if those carbohydrates are able to trap water and form these fantastic hydrogels, why not try to make the same, but instead of putting water, putting oil? So those are the oleogels. Oleogels are liquid water with a solid aspect, because you trap them in these carbohydrates. So we developed a patent. This was part of the thesis of Cynthia Fontes, which is now at Chagas working, where she was studying how the structuring of those phycocolloids could be used to develop these fat substitutes. So at that point, CSIC was investing money in making some cool videos of patents. So that video was released, and we had quite a big impact with that. So then we managed to ask for this project, which was a proof of concept project about developing these ingredients for substituting saturated fats in foods. And well, we had quite a big success. So we did some pilot trials with sausages just to substitute, and we managed to substitute even 100% of the saturated fat, and the sausages looked exactly the same. They tasted exactly the same. So we were okay. So let's keep on working on that. And we started interacting with some meat companies. They developed burgers, also substituting the fat with our oleogels, and they worked very nice. And what's the challenge? The challenge is the cured meat products. So in the cured meat products, consumers like to see the fat bits in there. So we are working in seeing how different oleogel compositions are able, while blending, to keep this aspect. That's it. And we are also exploring the use of those oleogels in bakery applications. So you need a different composition, because then you need to substitute butter instead of lard. And of course, as we have this relationship with companies from the seaweed industry, so we have this patent. It's the patent licensing option is signed for this industry, because we told them, look, you can use your phycocolloids also to produce this kind of ingredients. And as I told you, I love to explore other things. Well, in fact, in my postdoc, I was interacting with physics. I think it's amazing to interact with people that does different things, or that have expertise that it's completely different to what you do, because enriches a lot your research. So we sent Cynthia to a research state to a group that was working in biomedical applications. And they were interested because there are some chemotherapy treatments that they work first in vitro and then in vivo. So although there are systems that mimic some tissues that are water-related tissues, they didn't have any system to try on fat tissues, for instance, for breast cancer. So we thought, oh, maybe our oleogels are good for that. So she was developing formulations for testing them as fat tissues for these chemotherapy applications, and that was a very interesting research work. And they were measuring other type of characteristics that are needed, like dielectric properties, and adjusting these dielectric properties so that they could be used in other type of applications. And then, after extracting the phycocolloids, what is left, which is where we are trying to extract proteins and things like that, it's very rich in cellulose. We have a broad experience in extracting cellulose and characterizing cellulose and looking at it from different sources. So we thought, okay, so let's develop seaweed-based packaging materials using all the residues from the seaweed industry. In here we have done lots of things because it's also very interesting. The cellulose from seaweed, it's different from the plant cellulose. So it also has different properties. And we have, I didn't tell you before, but the first project that we had that we wanted to simplify the processes, we obtained less purified agar extracts, which were very interesting from a rheological point of view. They had polyphenols in there, so they had other properties like antioxidant properties, and we were, oh, we have found the best ingredient. They tasted horrible. Yeah, so they couldn't be used for food applications if you don't remove all the substances that provide this strong sea flavor, but these extracts can be used for producing films. In fact, there are companies that are developing agar films, but agar, it's very expensive to obtain material. So it's aimed in few proportions as a food ingredient, but it's also used for microbiological, so the agar plates and more advanced applications. But for making films, they are not competitive. I mean, plastics are very cheap, so you are not going to sell a food company, buy me these agar films. But what if we use these less purified agars to make films? So it's very interesting because agar is very hydrophilic. So if you put the agar in contact with water, it swells a lot. However, these less purified extracts, as they have other things in there, so proteins, polyphenols, which are interacting with the agar, then the functional groups which react with water are not available anymore, so they don't swell. So you can make films that then can be used, and in fact some of the properties are even improved with the presence of these other compounds. So that was a PhD that was fully funded by the company, by Hispanagar. So yeah, I thank Hispanagar for contributing to science, and she was doing lots of cool things related to developing, because these guys produce lots of residues and they were interested, why not biofilms from seaweed? Sounds like an interesting thing. So we have done lots of things. So understanding really how these kind of extracts behave as film materials. Also exploring different seaweeds. So depending on the seaweed composition, the films derived from these seaweeds behave also different. Also exploring if we extract the cellulose from this residue, how is this cellulose behaving? Also simplifying the cellulose process extraction. So with the cellulose process extraction, we did the same as with the phycocolloids. So what happened if we skip one of the purification steps of the cellulose? So we managed to reduce a lot the time. So from more or less 57 hours that are used in the paper companies for producing paper, for instance, they spend lots of water, lots of chemicals. If we skip some of these purification steps, we obtain cellulose, which is not that purified. You can see that it's not white, but it can have other properties. Cellulose, as I told you, is very hydrophilic, but it can also form gels. And from the gels, you can produce another type of gel, which is the aerogel. So we have seen the hydrogels, the oleogels, and now the aerogels. Aerogels are gels that the continuous phase, instead of being water or oil, is air, just air. So you just make the gel, and then you remove the water, and you have a very, very porous material, like the one you can see here. And what are those materials for? So those materials are very, very interesting because they are very light. They have a very, very high specific surface, so they can sorb lots of things, but they can be used also to release things. And they can have many, many different applications. There are companies commercializing aerogels. They are mainly used for isolating purposes, so to isolate tubes and things like that, but they are made from inorganic materials, mainly carbon-based, silicon-based materials. And we wanted to explore cellulose as an alternative organic-based aerogel. What's the problem of cellulose? That those cellulose aerogels had very poor mechanical properties, and they disintegrated because they were very water sensitive. There are research works working on hydrophobization, so you need to hydrophobize somehow to make it more strong and more resistant, but they are complex, they are expensive. And what we were interested in to keep the biodegradability of those materials. So we thought, let's think of a very simple method for that. So we patented a method that was very, very simple. Using a more hydrophobic biopolymer, which was still biodegradable, we just, by a dipping method, covered this cellulose with a hydrophobic polymer, and then those aerogels were water resistant, but their mechanical properties also improved a lot. So again, the CSIC made us a very cool video for that. And well, I didn't tell you, but this was the thesis of Isaac Benito. So yeah, they recorded a video, and they selected us in one of those accelerator programs to launch a spin-off company. So the spin-off company Aerofibers was launched. In fact, Isaac is now working at the company, trying to commercialize this kind of materials. And what has he been doing within the company? So he has been exploring different applications of these aerogels. So one very interesting of those aerogels is that when you apply this coating, this hydrophobic coating, if you immerse the aerogels in the mixture of water and oil, they preferentially sorb oil. So we thought, okay, for cleaning water. So for cleaning it, so he proved that it had this great ability for oil sorption, but also to be reusable, so that you can remove the oil to use again those materials. We tested them as alternatives for food pads. You know that in meat trays you have a pad underneath to absorb the juices of the meat. So we thought, okay, those are mainly covered with polyethylene. They are not very sustainable. Let's try our materials that are completely biodegradable for those purposes. And we proved that, of course, they can be used for that. They absorb lots of uses. But the good thing of these aerogels is that when you produce them, you can also incorporate bioactive molecules. So you can incorporate antioxidants. So what we did is to make this kind of pads introducing antioxidants so that the meat preserved the red color during more time, and lipid oxidation was also preserved. And we also tested as acoustic insulators because of this sorption ability. In a stay at Aalto University, he was trying those as acoustic insulators, and they were more efficient than the compound that is, or the material that is used nowadays, which is rock wool. And now we have a project to test them as I told you, that we can incorporate bioactives for release, so for biomedical applications as patches. So that's a project that we have with other CSIC institute and with the company. I'm out of time, maybe? Venga, 5 minutos. So very quickly. We also work with agroindustrial residues, in this case valorizing agricultural residues for extracting cellulose. Cellulose. Pectins. Pectins are also rheology modifiers. In this work, we also did lots of work in characterizing, studying how, in this case, it was watermelon rind pectin with very interesting properties as emulsifiers. So you can extract very interesting, and we understood how this pectin stabilize emulsions, and we test them with one company as an alternative ingredient in custards. And the last bit I'm going to show you is about fungal valorization. I don't know if you have seen this documentary from Netflix. If you haven't, please have a look at it. Fantastic fungi, amazing. I'm in love with fungi. So why are we studying fungi? Because there are lots of different edible mushrooms with many different compositions, many different properties, and we have used them not only to extract one carbohydrate that it's very, very interesting, which is the beta-glucans, which has also bioactive properties, but also because its residue is rich in chitin and also rich in proteins. So we are also doing, well, with the beta-glucans that we are extracting from residues, we are testing them as immunostimulants. So we have seen that depending on the structure of the beta-glucan, they can be like, they can prepare you for future infections. So they are very, very potent immunostimulants. And in collaboration with the University of Navarra, we are also evaluating, and I can tell you, eat oyster mushroom, because it has a glucose metabolism control, fantastic. So it helps you losing weight. And from the residues, we are also doing films. They are fantastic films. So depending also on the composition, the properties of the films that we obtain are different. Some of the films smell like chocolate, so they are edible films, which you don't need to add anything because the beta-glucan in there acts as a plasticizer. You don't even need to add a plasticizer, and they are completely biodegradable, as we have also tested depending on the source. So we are also exploring the Agaricus bisporus, el champignon, which is one of the most consumed ones, so the residues, the types of the mushroom, to produce films which can be used also in agriculture. Both the seaweed and these kind of films that are not very attractive for food packaging, but they can be used in agriculture and then be incorporated into the field, which can then help in crop production. And then just to thank all the research group, all the collaborators from national and international, of course, all the funding agencies, and all of you for your kind attention. Thank you very much.
Gracias, Amparo. Muchísimas gracias. Vamos a ver muchos éxitos en vuestro instituto y en tu grupo también. Vamos a ver, es el turno para vuestras preguntas. Supongo que Susana y Alejandro tendrán un montón de preguntas. Venga,
Por ahí.
Eduardo. Sí.
Thank you very much for your presentation. I have a question related with something that you presented during your presentation. It's the food, what do you call food viruses?
Yeah.
What is exactly food viruses? I mean, are viruses?
Norovirus, the norovirus. So there are viruses that cause diarrhea, so that are present in food products. For instance, hepatitis A sometimes is transmitted to humans through food. So there are all the viruses, the fecal, oral contamination viruses,
so
mostly.
So you are talking about human viruses
Yes.
that are in the food
Yes.
and are affecting
Yes,
the
that's
human
it.
population.
Yeah.
Now, my second question is related with the algae. You're working with a lot of algae and doing amazing things, amazing really. Where are those algae coming from? They are cultivated algae, or they are collected from the sea, or what is the main sources of those algae?
Industrial?
What do you mean industrial? They are cultivated?
No. Well,
The source?
Well, depending on the species, so there are seaweed species that can be cultivated, there are others that cannot be cultivated. So most of the algae we work with, they are just harvested. So, because I've learned a lot during these years, so there's a legislation telling how many tons of algae you can collect so that they grow again to make it sustainable. So the companies, I think that they pay for for collecting. So you have to pay a tax. So depending on your processing capacity, you collect many tons of. So we all obtain it from seaweed companies that they are using them for producing agar, alginates, carrageenans.
My last question is, what is an algae company working in Burgos? Why is in Burgos?
Agar.
Yes, but why in Burgos?
Why in Burgos? That's a very good question. I also asked them because I was like, you are not close to there. In fact, they were in the Cantabric Sea, but I don't know exactly why they moved the company to there. So, in fact, most of the algae they work with is from close to the Moroccan area, but also from Chile. So they get lots of Gracilaria from Chile. In there they are cultivating because they work mainly with Gelidium. So they are both red seaweeds. So red seaweeds are used for agar production. And the Gelidium cannot be cultivated, so they have to be harvested, but the Gracilaria, they manage to cultivate it, so most of the yeah, the resources are coming from Chile, in which in there it grows like crazy.
I have one comment to your comment, which is that, well, probably you know, I was born and grew up in Cantabria, which is very close to Burgos. And when I was younger than now, nearly 50 years ago, all children in my village, Laredo, we went to the beach for harvesting algae.
Yeah?
Yeah,
Yeah.
and price was 5 pesetas, 100 kilograms. So you can imagine, paid by the pharmaceutical company.
Yeah.
And it was the way of getting the old children la paga.
Claro, claro. Yeah, yeah, there's a whole business around seaweed and collecting seaweed and things like that in that area. In fact, there are lots of seaweed companies around the north of Spain working with algae and cultivating algae.
Now, I think that such activity, as you said, that now is in the northern part of Morocco and Chile, probably the children in Laredo do not go anymore. I mean, they have to be contract and they have to have insurance and things like that. Okay, Pablo.
Hi. So I have a question. So of course here you don't have to explain that how important this is for ecology and stuff, but the question goes for industry and the value of reinventing the wheel, because many of these materials already exist in another form that are way cheaper, and industry, you know how it works. Unless there is a legislation that pushes them to go away from plastic, they will still use plastic.
Absolutely, yeah, I completely agree. I mean, in fact, I didn't show you, but I have also a kind of an ethic problem with bioplastics, because I mean, I'm not demonizing plastics, because I think that plastic is a fantastic material. In fact, I'm from the plastic. My PhD was on plastics, and I think it's great. But you need to use plastic for not for what? Joder, perdón. Single use, not for single use things, because it makes no sense. But there was a legislation saying that straws, so these plastic straws and certain commodities, plastic commodities, have to be banned. Because they contaminate a lot, so they have to substitute it. What was the result of that? So there were lots of companies, lots of bioplastic companies. Those bioplastic companies, they are developing biodegradable materials. What are biodegradable materials? Those OK compost things that we find in supermarkets and things like that. So it just means that it complies with regulation. Okay, so if you put them in certain conditions, they degrade in X days. And they comply with the toxicological analysis. What's the toxicological analysis in this kind of norm? That if you put this material and you grow gramínea, it grows. That's all the toxicological analysis. So a colleague from other CSIC institute contacted me once, and she told me, because we have at IATA, we are part, Susanna, and they are also part of the SUSPLAST platform, which is one of those interdisciplinary platforms from CSIC. And she was also from SUSPLAST, and she told me, oh, I've seen that you have, from SUSPLAST, a composting unit at IATA. So according to norm, we are able to characterize if the materials are biodegradable, compostable, or things like that. And she was interested in understanding what was the toxicity of those new commercial bags. And I was like, oh yeah, okay. So just send me the materials, we will characterize the composition, of course, the macromolecular composition, because I knew which polymers are used in these compostable bags that we all found in the supermarkets, but I don't know the additives. And there are, I don't know how many additives that can be used for plastic processing. I mean, that was also a cornerstone in food science, because when food technology developed, they were using additives that are then banned because they found that they were toxic. But they need to put in the food, in the labels, what are the components that you are using for making your food. But they don't say what they are using for making the plastics or the bioplastics. So what was the surprise? So yeah, we characterized the material, we made the composting, we sent them after composting the material back so that she could analyze the toxicity in some models, cell models that she had. And when she sent me the manuscript, so a draft of the manuscript, I was reading and I was like, oh my God, carcinogenic, they are more toxic, than the plastics, than normal plastics. So we analyzed a normal plastic bag, so a polyethylene plastic bag, and then a recycled bag, and then the compostable bags. Or surprise, the compostable bags, the more toxic ones, and after composting, even more. What was our hypothesis? That yeah, they are using some additives that are toxic, and when you decompose the material, so they are releasing those. So yeah, we launched one of those press notes. Oh my God. I was amazed because all the bioplastic companies came to tell me that, yeah, I was trying to make us say that our experiments were wrongly done, that this is not a real situation, that we extracted with methanol. Methanol is not in the environment, and it's all a cleaning thing. So when you put a new norm, like banning this kind of items, you need to be sure that you have a secure or safe alternative to commercialize. Otherwise, they say, we comply with the norm. It's true, they comply with the norm, but those materials are not good, and probably, industry works at a different path. So science is like a more slow path, that we cannot go as fast as it is really needed. So what we are trying is to develop compositions, which of course are not nowadays competitive from an economical point of view. That's why we are trying to simplify processes to make them economically viable, and trying not to use additives that can be toxic to the environment. So not solving a problem and creating a bigger one. Because at the end, the problem of all those compostable bags is that they have to be thrown in the compost because they go to composting plants. But who knows where people throws, so they will end up in the environment and they will cause problems. Because I have a colleague that has also a small composting plant, and because I had lots of stress at that moment, because I had lots of pressure from the companies, and he was telling me, I can tell you that you didn't do anything wrong. When I evaluate the composting properties, the worms that are in they die. So there's something that is not good in this kind of materials.
Hello, Amparo. Nice talk. I'm microbiologist, so I think, yeah, I feel like I am more like 80% microbe than human. But
more than 50.
Well, my question is, you mentioned that seaweed has also a lot of you call residues or bioactive compounds.
Yeah.
Do you know the proportion of these bioactive compounds that are coming from the microbiome and not from the seaweed? And
No.
No, okay. Well, my next question was, do they have any pharmacological use? That's, yeah.
I think that nowadays, well, at least I'm not aware that there are seaweed extracts that are used for cosmetics, but I think that there's lots to do, because depending on the species, the composition is different and the bioactive presence are very different. For instance, certain brown algae, they have fucoidans, which are certain carbohydrates, but that have kind of polyphenols strongly bound to them, which have lots of bioactive properties, but they are not exploited commercially yet. So there's lots to do in that way, and I'm sure that many bioactives are produced by bacteria. And that's another point very interesting, because the bacteria, as we humans, you like certain things and you don't like certain things. So depending on the bacteria, they preferentially, because they have enzymes to hydrolyze certain compounds, so depending on the bacteria, there are certain biomass that have more of a certain kind of bacteria than others, and that you can also use bacteria to extract. So that's another thing in which we are collaborating with other groups that are more experts. Well, more experts. We are not experts at all in microbiology, but that can use microorganisms as a way of extracting compounds, which is greener than using organic solvents.
Thank you.
Yeah, Mark, more questions? One question, what about, well, many years ago I was working in the starch field, and
Me
it
too.
was, yeah. Yeah, at that time it was the heat, no? That to use starch as a raw material for bioplastic production.
Yeah.
How is it now, this subject?
Well, in fact, many commercial bioplastics contain some starch. But what I'm saying is, starch is a very rich nutritional source. I wouldn't use starch for making films, because starch is for eating. I mean, we are going to have problems in having foods for everyone, so I would rather use something that it's not used for anything else for making films than something that you can eat. You first eat it. If you cannot eat it, then use it for packaging. But starch is a fantastic material for processing, of course.
Yeah.
Yeah.
Okay, more questions? No more questions? So thank you very much again.
Thank you.
Muchas gracias. And now...
Boom, boom, boom,